Prevention of antiviral drug resistance. Strategies to prevent the development of antiviral drug resistance include the judicious timing of treatment and patient education regarding adherence to therapy. We know these are important initiatives in terms of the relationship between the physician and their patient. In the context of first-line therapeutic choice, high potency drug with a high genetic barrier to resistance, such as entecavir and tenofovir, should be considered. Pegylated interferon still has a role as an alternative first-line therapy if the patient, especially if the patient has high ALT and low HBV DNA. These are the hallmarks of a good responder to interferon therapy. In the context of monitoring, regular 3 to 6 monthly monitoring for viral load with a sensitive WHO-validated HBV DNA assay that reports in international units per ml. And genotypic resistance testing should be considered in those patients with their first and subsequent virological breakthrough. For salvage therapy, early initiation of add-on salvage therapy is highly recommended. The avoidance of the switch sequential monotherapy and the use of combinations of drugs with similar cross-resistance profiles is actively discouraged. In summary, the current emerging patterns of antiviral drug resistance to the hepatitis B virus polymerase are complicated, but 4 major pathways can be defined for the primary resistance pathways and a 5th multi-drug resistant pathway. Primary resistance mutations across the nucleoside-nucleotide analogue groups has been identified through the A181TV substitution, as have broad clusters of compensatory mutations during lamivudine long-term monotherapy, and that these compromise future salvage therapy options with the newer agents such as entecavir. There has emerged definitely the need for or the requirement for HBV polymerase sequencing to determine the profile of drug resistance, and we've highlighted the emergence of multi-drug resistance as a clear cause for concern for physicians. The public health issues around the fact that a drug-resistant virus does have an altered envelope because of the pol overlap, and the implications in terms of transmission and possible vaccine escape are areas for future concern. The opportunity and the need now for, in the future is for newer antiviral agents targeted to other sites in the viral life cycle are clearly needed and really essential if we are to eradicate hepatitis B virus from our patients.